Assalamualaikum and greetings everyone. Welcome to our lab session on fish dissection and observation. Our group members consist of Maizatul, Amanina, Sharil, Hani, Christina and Ersiana. The objective of this lab is to provide us students with hands-on experience in fish dissection, allowing us to observe and understand the external and internal anatomy of a fish. This session aims to enhance student skills in anatomical identification, organ function analysis, fostering a deeper appreciation for biological structure and function while emphasizing the importance of laboratory safety and precision.
Before we begin, it's important to have all our materials ready. During this lab, we use a dissecting kit, gloves, tissue dissection tray, a fish specimen, ethanol 70%, ethanol 99%, water, trooper, light microscope, glass slide, cover slip, marker, alcohol lens, and sample bottle. We also carry out this study by following all safety guidelines throughout the process. We start by laying out our materials on a clean work surface and putting on our gloves. Then we place the fish specimen on the dissection tray, positioning it with its dorsal side up.
After that, we began observing the external features of the fish. We noted if there were any abnormalities for the head, caudal fin, dorsal fin, pectoral fin, anus, skin, eyes, gills, and mouth. These steps helps in understanding the basic anatomy before making any incisions.
Next, using a scalpel, we made an initial incision along the ventral side of the fish, starting from the anus and moving towards the head. While cutting, we were careful not to cut too deeply to avoid damaging internal organs.
Then, we spread the incision to expose the internal organs. We identified and observed the major organs such as the heart, liver, spleen, intestine, kidney, and brain. During observation, we took our time to examine each organ and make notes whether each organ is normal or abnormal.
After completing the dissection, we clean our tools and workspace thoroughly, dispose of any biological waste. And that is it for this lab section. This process helps to gain valuable insights into fish anatomy and prepares us for further studies in biology.
In this session, we will be showing two essential microbiology techniques, the strict plate method and gram staining. These methods are fundamental for isolating and identifying bacterial species. Let's get started. First, let's discuss the strict plate method. This technique is used to isolate a single species of bacteria from a mixed sample. Here's how it works. Step 1. Sterilize the loop. We begin by sterilizing our inoculating loop in the flame of a Bunsen burner until it is red hot. This ensures no contamination.
Dip it into the bacterial culture to collect a small amount of bacteria. Carefully streak the loop across the surface of the agar plate in one quadrant. Sterilize the loop again, then streak through the edge of the first quadrant into the second quadrant. Repeat this process for the third and fourth quadrants, sterilizing the loop between each strip.
Once tracking is complete, incubate the plate at an appropriate temperature for 24 to 48 hours. After incubations, we discover isolated colonies that have grown from individual bacterial cells.
Now, let's move on to gram staining. This differential staining technique allows us to classify bacteria into gram-positive and gram-negative based on their cell wall structure. First, we started by preparing a bacterial smear on a clean microscope slide. Air dry the smear. Then heat fix it by passing the slide through the flame. Next, we flood the slide with crystal violet stain and let it sit for 1 minute. And then, rinse gently with water.
After that, we apply iodine solution and let it sit for 1 minute. This acts as a mordant, forming a complex with the crystal violet. The next step was decolorize the slide with alcohol for about 10 to 20 seconds. This step differentiates between gram-positive and gram-negative cells.
Step 5. Counterstain with safranin. Finally, counterstain with safranin for 1 minute. Then rinse with water and we let it air dry. Step 6. Examine under microscope. After the slide is dried, we examine the slide under a microscope. Gram-positive bacteria appear purple, while gram-negative bacteria appear pink or red.